In clinical practice, esophageal stricture occurrence is a major limitation for extended endoscopic resection of esophageal superficial neoplasia. This video presents an ADSC-sheet transplantation technique that prevents such strictures, making extended endoscopic resection possible in a porcine model.
This method can help answer a key question in the field of esophageal stricture prevention after endoscopic extended resection. The main advantage of this technique is that endoscopy ADSC-sheet transplantation is minimally invasive, effective, and not time-consuming in order to prevent such esophageal stricture. Demonstrating this procedure will be Gabriel Rahmi, a physician from my laboratory, and myself.
Collect around 24 million ADSCs per pig using standard cell expansion procedures and T150 culture flasks. Harvest the ADSCs using 10 milliliters of standard trypsin solution per flask. Then, count the cells using an automatic cytometer. Load 1.5 million cells into each well of a 12-well temperature-responsive cell culture dish. Now, top up each well to 4 milliliters with culture medium. Then, incubate the plates for 12 hours overnight for the transplantation on the following day. On the day of the transplantation, detach the confluent cell sheets in the plate wells by incubating them at room temperature for 30 minutes. After half an hour,